Hello, Miss. How are you? Yeah, let's start. Okay, because in this session نرجع مثل overview على ال translation mechanism. حنحضر 2 remaining animations بتلخص ال transcription translation مع بعض. And بعد في فكرة صغيرة بدنا ناخدها and then we will write at the end of the session. So نحن حكينا عن ال steps of translation. Initiation, elongation, termination. As you can see in the figure, initiation starts when the small ribosomal subunit with large, they bind together, they scan the messenger RNA looking for the first AUG. AUG will be situated facing the P site. The transfer RNA يلي هو complementary to the AUG حيجي يعمل bind على ال AUG carrying the amino acid met. The A site is empty. In the elongation process, the transfer RNA يلي هو complementary for the second codon will come and fill the A site, as you can see in the first figure. Then a peptide bond will be formed between these two amino acids. You should know that this peptide bond is more powerful than the bond between the transfer RNA and the amino acid. So حتقشطوا إياها لtransfer RNA. So the peptide bond now is formed. It's time for the ribosome to translocate, to move one codon. It's time for the ribosome to move one codon. As it moves one codon, transfer RNA اللي كان بالA بالP site will be left behind. هيدا transfer RNA, we'll watch it in the animation now, will be left behind. حيصير برا. And the one that is found in the P site, A site اللي هو قدام, حيصير هو حيندفش لل P site. The A site is empty again, okay, to be filled by another transfer RNA that is complementary for the codon. This continues until reaching a stop codon. When the stop codon is reached, it means the end of translation. So when the stop codon is reached, as you can see, the peptide will be released, the methionine will be liberated, will be removed, and the small ribosomal subunit and the large ribosomal subunits, they will dissociate, they will separate from each other. Do you have any question about translation before we continue? Do you have any question before we continue? Okay. Some of you joined late. Noor, Muhammad. Yeah. Let's continue. هنحضر هلا animation. هنحضر animation. نحن امبارح حضرنا one animation. Watch the other one. يلا ركزوا فيها منيح. هيدي خاصة بالميكانيزم of translation, بس حتفرجيني عملية translation. Okay?
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific.
Okay, البولي A tail هو من جهة ما بعد الستوب كودون. يعني هو part which is not synthesized where the protein is not synthesized. نحن قلنا reading frame starts from the AUG and ends with a stop codon. So هيدا حيكون ما بعد ال stop codon. هي قلت لكم هي protection sequence بتكون، وقبل start codon كمان أنا عندي protection sequence كمان it will not be synthesized. Yeah, let's continue.
Amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter.
شايفين كل ما بتمشي أكتر الرايبوسوم على المسنجر RNA, the length of the peptide will increase, as you can see. Okay, and this transfer RNA اللي كان بالP site will be left behind, حينترك, and the one that was found in the A site قدام حيصير ورا ساعتها the A site will be empty, ready to receive another transfer RNA.
When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay. Yeah, another animation. You can hear the sound and see the video. Yes?
Yes, teacher.
اوكي، لا انا بعد ما سمعت صوتكم. يلا.
DNA is very long and cumbersome. The strands of DNA also...
Okay, this animation you will see the transcription. هترجع تذكرني بالtranscription, how it occurs from DNA to messenger RNA. هترجع تحكي أكيد عن translation and the mechanism of translation. Okay, so انتبهوا هيدا تلخيص يعني كأنه activity 3 and activity 4 together.
the code for many different proteins. To get the code for the desired protein from the nucleus to the ribosomes in the cytoplasm, a copy of the small necessary section of DNA will be made. The copying process is called transcription, and it occurs in the nucleus. It's the first step in protein synthesis, the process of making protein. The second step is called translation. A portion of the DNA is unzipped so that the mRNA can be made from the DNA like a template. That's transcription. Nucleotides of RNA match with one strand of DNA and make mRNA. RNA polymerase unzips the DNA and puts RNA nucleotides into the right place. C and G nucleotides match up, A and T nucleotides match up, but anytime the RNA places a complement to an A on the DNA, it places a U instead of a T in the RNA. Remember, RNA uses uracil instead of thymine. So, if the DNA strand looked like this, the complementary mRNA strand would be the complement, but each time there would normally be a thymine, there's a uracil in its place. The mRNA also doesn't copy the entire strand of DNA. It only copies the portion of DNA that codes for the protein it wants at the time. In fact, most of DNA doesn't code for anything. Only about 2% of it codes for protein. So the small section of coding DNA has a start and stop signal. After the mRNA is formed, it moves out of the nucleus through nuclear pore and goes into the cytoplasm. Remember, there are 3 different types of RNA. The purpose of the mRNA is to get the protein code out of the nucleus without pulling the DNA out of the nucleus. Then the mRNA will need to find a ribosome in the cytoplasm. Ribosomes can be found on the rough ER or floating in the cytosol. mRNA is read 3 bases at a time, and these 3 bases are called codons. Now, the ribosomes are made of protein and RNA, and the ribosomes are the protein-making machines that read the mRNA code and add the correct amino acid using tRNA. Remember, tRNA stands for transfer RNA, and that's because its purpose is to transfer the right amino acid to the ribosome to build the protein. The tRNA has an anticodon on one end, which will match a specific codon on the mRNA, and has a specific amino acid on the other end. Together, these 3 parts will make translation happen. Translation occurs in the cytoplasm of the cell wherever there's a ribosome. To get translation started, mRNA attaches to a ribosome, and a start codon must be read. It's usually AUG, and I remember that school starts in August, and that helps me remember AUG. The first amino acid is brought in by tRNA. The anticodon on tRNA matches up to the codon on mRNA. Then the next tRNA molecule moves in and matches up with the mRNA codon. This time, the amino acids form a peptide bond and link together. Then, the first tRNA can detach, and the mRNA shifts through like ticker tape, and the next tRNA molecule can come in. The protein grows until a stop codon is reached. Once the stop codon is read, the protein is formed and ready to finish folding to become functional. And that's the end of protein synthesis. Now, we can figure out the sequence of amino acids using the mRNA and amino acid chart like this one. Let's use an example. Starting with the first 3 letters, AUG, we can find the amino acids. The first letter is A, which narrows it down to this row. The second letter is U, which narrows it down to this column. And the third letter is G, which means that this is methionine, which is the starting amino acid. Next is CCC, which is the second row, second column, and second row on the right. But we don't even have to look at the third letter in this case, because every third letter will bring the same amino acid, proline. Next is GGC, fourth row, fourth column, second row on the right. That gives us glycine. And lastly is UAA, row 1, column 3, row 3, and that gives us stop. There's no amino acid here. It just means that the protein will detach, and it marks the end of protein synthesis. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on Twitter @SciencePet.
Okay. So حننتقل نحن على شيء اسمه poly. Yes. Any question?
لا بس بدون كان بعد في انيميشن نحن كانت معلقة على الصورة ما حضرتينا اياه. هو هيك في 2
إيه ما بقص. ايه عرفتهم. إذا بقي وقت بحطها آخر الوقت. أوكي؟
افتكرت ناسي مشكلة.
إيه إذا بقي وقت. إذا لا بشوف إذا ببعتها لينك تحضروها أنتوا بالبيت. طب let's see who joined. كمان عنا حسين سكيكي. Yes, joining late يا حسين. ومحمد صالح also late. Yeah. إذا آخر فكرة عنا هي polysome. What is a polysome? Or call it polyribosome. It's polysome or polyribosome. من اسمها poly, the word poly means many. So it's many ribosomes. Many ribosome. Okay? It is the same messenger RNA as you can see in the figure, where many ribosomes are moving along the same messenger RNA. يعني أنا عندي كتير ribosomes عم بيمشوا على نفس المسنجر RNA. This is called a polysome or a polyribosome. Okay? Let's say I will give you now each ribosome is capable of synthesizing one protein. So each ribosome is capable of synthesizing one protein. If I have 10 ribosomes moving along the same messenger RNA, how many proteins will I obtain? Okay, how many proteins do you think? If each 10. Okay, if I have 10 ribosomes, then I will obtain 10 proteins. Are these proteins identical or they are different?
Of course, different.
But I'm translating the same codons. How come they become different? إذا أنا عندي ورقة ومرق عليها 10 مترجمين، مش كلهم لازم يترجموها نفس الشيء؟
Yes.
It should be identical because I have the same codons، والcodons أنا عم طلعهم من الجينيتيك كود تيبل، هن ثابتين ما بيتغيروا. كل كودون عنده specific amino acid, so it will give me the same or identical proteins. نحن عم نحكي on the same messenger RNA. يعني عم بحكي على نفس المسنجر RNA. So polysome or polyribosome, okay, هي the group of ribosomes that are moving along the same messenger RNA. They are translating the same protein. أكيد عم يعملوا translation لنفس البروتين. For example, if I have 10 ribosomes moving along the same messenger RNA, then 10 proteins will be produced having the same amino acid sequence. So these 10 proteins should be identical in their sequence. Okay. أنا هلا بدي أرجع شوي للsteps of translation. Translation requires energy. Okay, so translation needs energy. What do you think, in your opinion, which is the step of these three, which step out of these three requires the largest amount of energy?
Elongation.
اوكي elongation. ليش برأيك عبادة؟
مشان بمرحلة الالونجيشن عم بيصير انه البي سايت عم بتروح عم يجي بمحل عم بضل انه يتكرر
اوكي
بروتينز وهيك.
في two important steps بيصيروا بالelongation تتذكرون؟ في شغلتين كتير مهمين بالelongation. أنا بعرف إن إحنا بعد ما كتبنا.
بصير في replacement.
translocation. بصير في translocation صح؟ يعني the ribosome moves one codon. This requires energy. This step needs energy. وشغلة تانية بتصير بالelongation كمان requires energy. So I have the translocation of ribosome. What else? Can you remember? formation of what؟ حط الرسمة أنا بركي بتذكركم بشي. Two important steps occurs in elongation. لما يصير transfer RNA هني occupied ب both sides لازم يصير في عندي what؟ Formation of
peptide bond.
peptide
bond. So the peptide bond formation and the translocation of ribosomes هن سبب إن تكون elongation most requiring energy step in the protein synthesis. So بيطلع عندي هون the peptide bond هذا أول سبب. And the translocation of the ribosomes during the elongation step of protein synthesis requires the most amount of energy. So هي ال most requiring energy step بالprotein synthesis. Okay? Okay, I have to switch. أنا راحت الكهرباء. يلا أنا حابعت هلا الفايل بس خليكم معي because I need to make sure that about the attendance لأن في ناس راحت في ناس اجت. Okay.